Okay, hello everyone. Welcome for another seminar the EBD seminar series. Today we are having Noé Fernández, who is a Ramón y Cajal researcher in plant genomics. He has a long and very international trajectory after doing his PhD at the University of Malaga with Professor Gonzalo Claros. He has worked for at least 5 years as a postdoctoral researcher and research associate in the Boyce Thompson Institute, which is a research institute associated with Cornell University in New York, United States. Afterwards, he has been working as a research assistant for at least 4 years at the University of Marburg in Germany. And then in September 2021, he joined the La Mayora Finca, if I'm not mistaken, at CSIC La Mayora Finca, where he is working as a Ramón y Cajal right now. He's now here. Thanks to Teresa Boquete for giving us his contact. And we thank you very much, Noé, for being here and giving us your presentation. Please go ahead.
Okay, good afternoon, everyone. Thank you, Agustin, for this nice introduction. And first of all, I would like to introduce a little bit my institute, because most of you might not know it. This is the Institute of Subtropical and Mediterranean Horticulture, which is in Malaga, in the southern Spain. And we recently have now a brand new building on the campus of the University of Malaga. And of course, we have our nice experimental station in Algarrobo, also in the province of Malaga, where we have 50 hectares of land to grow subtropical and Mediterranean crops, such, for example, some nice avocados and mango we have here. And the Institute is known for introducing strawberry crops and subtropical crops in the southern of Spain. And as Agustin said, I recently joined the Institute in September. And something that surprised me is, like, after 9 years abroad, when I came back to Spain, many people that I met told me that they have used the tools I've developed. So it's something very nice to hear. Some of these tools that I will tell you about today are, for example, the Soil Genomics Network. That is not a tool I developed myself. I didn't create that because it has already a few years. It was developed by Professor Mueller, but I was working there for 5 years. And other tools that I actually developed are, for example, the Tomato Expression Atlas, the SGN VIGS Tool, Olive 3DB, or Pitmos. The Soil Genomics Network, for the ones of you who don't know it, is the main genomics portal for soil analysis. These are, for example, tomato, potato, pepper, eggplant, tobacco, and Nicotiana benthamiana. The soil genomics have a lot of bioinformatics tools, and it hosts a lot of genomics data, but the system is also something we can reuse in other genomics portals. For example, there's another implementation in citrus greening solutions, where we have data of the citrus greening disease. We have data from the host, the vector, and the pathogen. And now recently the SGN, the system have evolved to breed-based, and now it's possible to manage a breeding program. And there are many popular implementations, for example, such as cassava-based. But I'm not going to talk much more about the Soil Genomics Network, and I will focus on other tools, for example, the Tomato Expression Atlas, which I'm very proud of because it gave me the chance to work with great researchers of Cornell University and the Boyce Thompson Institute. And for this project, I developed this 3-dimensional cube where we can visualize the expression data. And I also created other visualization methods with cartoons of the plant and others. But the most amazing of this tool is actually the data that it hosts. We have an amazing data set of the development of the tomato fruit, of the fruit ripening. We have stages from the anthesis to the red ripe stage. And on top of that, for the mature green, the breaker, and the pink stages, we have 3 cuts of the tomato. We have the top, the medium, and the bottom part of the tomato. So in general, when people study the tomato fruit, they usually only study the whole pericarp. But in our case, for our data set, on top of all these many stages of development that we had, we also studied the pericarp, but also we included seeds, locular material, septum, placenta, and columella. And additionally, for the pericarp, we got data for the whole pericarp, but also we use laser capture microdissection to separate all the different cellular types of the pericarp and have a very high resolution on it. And we have data for the outer epidermis, the collenchima, parenchyma, vascular tissue, and the inner epidermis. So all this data can be visualized in this three-dimensional cube where we have on the top all the stages of the development, and in this diagonal we have all the tissues with the gray background for the laser capture microdissected data. And here on the left in blue, we can see our query gene, the gene which we want to see the expression, and below we will see co-expressed genes. Genes that have a similar profile of expression and might have similar functions. But of course, you can introduce your own custom list of genes and visualize them in the cube, or also we have an integrated BLAST tool, and you can use the result from this BLAST to visualize the expression data in the tool. And this is more or less how the output of the tool looks like. On the top, we have the query gene with a link to the Sol Genomics network where you can get all the information about this gene. You can search for another gene or play with the scale, the color scale, in case that you have, I don't know, expression data that you want to have high resolution in the lower values or in the highest value in case you have some saturation. We have multiple visualization methods, such as the expression cube, expression images, heat maps, scatter plots, DEGs, co-expression network. We can download the data that are shown in the cube. We can download also its replicates. Of course, in the input, we can filter which stages and tissues we want to visualize. And this is a very interactive tool. So you get an idea as you move the mouse over the gene names, you can see the description of each one of the genes and the co-expression values. So that way, in a very fast way, you can visualize all the genes that are co-expressed under the definitions. You can click on the genes and you can split the layers of the cube, or you can click back if you want to collapse them back. That way you can compare multiple genes and explore their expression. You can also click in any of the layers of the cube, and that will open a bar plot graph. That way you can open as many bar plot graphs as you want, and you can visualize the stages and the tissues in the x-axis or in categories, and you can transpose that, which make very easy to compare the data. In this case, we have a lot of stages, but as I said, you can filter them in the input in case you want to focus only in a few of the stages or tissues. Some nice things that also we get with this great data set is like the co-expression of the genes is great. For example, in this case, if we see the expression of a chalcone synthase, we can see that most of the co-expressed genes here on the left that are in red are part of also the flavonoid biosynthesis pathway. And one of them is also one of the transcription factors that control many of the genes in this pathway. So that can give you also some nice information about genes that are co-expressed and you didn't consider, for example. Additionally, as I said, in most of the studies, for the fruit, usually only the pericarp is studied. In our case, as we have all these other tissues of the fruit, we obtain another 2,400 genes that were expressed in the fruit, but not in the pericarp. And additionally, with the laser capture microdissected data from the pericarp in detail, as some of these layers of cells are very, very thin, it's possible that the expression of the genes get diluted with the rest of the tissue, so they are hard to get in your results with the total pericarp. But with the laser capture microdissection, we were able to find more than 1,000 genes that we could detect it in all these tissues, but not in the total pericarp. Some nice examples of how we can see over the development of the stages and the different tissues, how the genes are expressed are, for example, for genes associated with fruit softening, we have the PG2A that we see in the breaker stage. It's like basically the expression starts outside of the fruit, and then we see how it gets higher in the rate ripe, also mostly in the external part. But in other example, like PL1, we see the opposite. This in the mature green, that is a stage where the tomato is still green and hard. We can see that the expression of the gene is already starting inside the fruit. And then from the inside in breaker is starting to go to the outside. And in the red ripe, we already see a high expression almost all over the fruit. Some other examples of very specific expression data are for these two genes associated with flavor. In the case of ceton T1, sorry, in the red ripe stage, we can see that this is specifically expressed in the vascular tissue. And in the case of TOMLOC-C, in the red ripe stage, we can see that it's mostly expressed in the collenchyma. So these are very nice examples of the high resolution of this data set. Additionally, I would say this tool was quite successful, so we were implementing that in other projects that also require something similar. Some examples of that are PIDMOS, where we have RNA-seq expression data and expression microarrays for fiscomycium patents. We have also an implementation for the citrus green disease. We have data for the host, the vector, and the pathogen. And in this case, we have some data sets which are from RNA-seq and other data that are from proteomics. In the same way, in another private implementation of the tool, we have metabolomics data. So the tool is very flexible and it can host data of all kinds of types. And a special mention I would like to do about this implementation is PIDMOS, where we developed a expression atlas for the model of fiscomycium patents. That was the model of the bryophytes, was the first genome of bryophyte sequence in 2008, but it had many gene versions over the years. So when we started this project, there were like maybe 3 or 4 platforms with some expression data, just a few that have data in many different gene versions. So if you wanted to search for the gene expression for one gene, first you need to know in which tool do I have to look for this data, which gene model version I need to choose to query this data. And then we unified all this data in one single tool with more visualization method than all the others together. We included many new data sets, and now we provide over 100 experiments where we have the whole life cycle of fiscomycium patents, and we also have many experimental conditions. Another tool that I would like to present is the SGN VIGS tool. The SGN VIGS tool is a tool to help the researchers to design their construct for virus-induced gene silencing. Virus-induced gene silencing is basically the mechanism that the plant have to defend itself from attacks from viruses. When a virus is introduced in the plant, they start to reproduce their genes in a very high rate. So the plant has some mechanism to stop the translation of these genes, to silence these genes. So the same way we can use this to our advantage in the lab, and we can introduce also double standard RNA using, for example, a vector such as agrobacterium, as the virus does. And then this double standard RNA will be detected by dicer, which will digest these messenger RNAs. Sorry, the double standard RNAs will be digested into small RNAs of a size of 21 to 24 nucleotides. And that this small interference RNA will be recognized by the complex RISC that will use them as a template to identify the messenger RNAs that, for example, could belong from the virus and digest them so they cannot be translated to proteins. So let's say that RISC is more or less like a policeman that is checking for the ID for each one of these messenger RNAs and checking if they come from the virus. So this process was discovered in an experiment in the 90s where they were trying to overexpress the chalcone synthase gene in petunia flowers. They wanted to get very deep purple coloration of the petals. But instead of that, what they got was white flowers or partially white flowers. And that was because as the gene was expressed on to a very high rate, this transgene, the plant detected that and suppressed the expression of these genes and also the endogenous gene of the plant. So resulting to get white coloration instead of a deep purple. So the idea of the tool is the same. It's try to replicate in the algorithm basically what happens in the cell. So it will try, you can paste just the sequence of your gene, and the tool will try to identify this gene. It will be split in small interference RNAs, and we will identify all the possible messenger RNAs that will match to that. And we will detect which ones are the best targets, the genes you want to silence, avoiding to silence any other off-target genes. When we developed the tool, in the first version, we have only 9 species data sets, but eventually many researchers start asking for more and more data sets. And today we have over 50 species data sets. The overview of the output of the tool looks something like that, where we have a control panel and a graphical overview where we can, on the fly, we can play with the design of the construct for the VIGS. And then below we will have the FASTA sequence of the construct and the position in the gene. And at the bottom, we will have the number of matches we got for each one of the genes. So you get an idea. Basically, the tool will predict the best region in yellow and will show you what is the score for that region. You can create your own custom region, and also you can check the score and move it and resize it to get the best of it. In blue are shown the target genes, the genes that you want to silence. In this case, there are two because it's in Nicotiana menthamiana, which is a polyploid, but in many cases you might have only one copy. And in red are shown the red genes, the off-targets, genes that you don't want to silence. These are not part of your experiment and you don't want to have a side effect in your phenotype. And at the bottom, you can see a score graph that is telling you which are the best regions, the regions that cover the most your target genes and are avoiding the off-target genes. So we can expand the graph to see in detail each one of the small interference RNA that align to the gene. And at the bottom, you can see the total number of them. And additionally, we could provide also an expression file where it will show the expression of each one of the genes in the experimental conditions that you have. So that will help you also to evaluate if that gene is actually expressed or not in your experiment. So you get an idea. Also, this is a very interactive tool. You can set your own custom region here, and you will see at the bottom it will appear the FASTA. You can move the custom region, and as you move, it will update at the bottom the sequence. You can resize it, and it will update again the sequence and the position in the overview of the gene. And as you move it, of course, you will be getting different scores. You can expand it to see in detail the amount of small interference RNA. In this case, we have a very bad score because we have a lot of targets in red. And in this region, we can zoom in to adjust perfectly to the extremes, to the borders of the region we want to silence. And once we have it, if we get a better score, we can use that to design our VIGS construct. But now, last year, I recently joined to the IHSM, the institute that I mentioned in the introduction. And in this institute, we are studying subtropical fruit crops on top of some Mediterranean ones. And here we have a very amazing collection of subtropical fruits. This collection probably is the best one in continental Europe, where we have over 300 accessions of cherimoya, which is the largest germoplasm bank of cherimoya in the world. We have over 100 accessions of avocado, and we have around 80 accessions of mango, and many more from the other species. So we have really great resources to learn more about these species and to improve the genomics of them. The thing is, of course, these species are subtropical, so they are adapted to the conditions in the southern Spain, and their productivity is limited to the amount of water. But it's very nice that in our collection, as you can see here in the picture, we have a great diversity of sizes, color, shapes, flavor, also the time of the year where the fruits and the flowers are produced. So we should take advantage of that to get the best for our research and also for the commercial products of these fruits. The problem is in some cases is like in the market, for example, in cherimoya, 95% of the cherimoya are from the variety Fino de Jete. In avocado, 85% of the avocados are of the variety Hass. And in mango, in the southern of Spain, 80% of the mangoes are Austin. So there is not really a big variety in the market production, which is not nice for many things. For example, for the variety of the season of the year where the fruit are produced, but also in case if some disease appear, it's like it could attack to these cultures where are based mostly in one of the varieties. Also, these species are not easy to work with because most of them are trees, and they are non-model species. So a few years ago, there were not even genome sequences for any of them. And now that we have some genome sequences, there are poor annotations for the genomes, and there are not many bioinformatics tools that we can use to use this information. However, I don't know if you know, but recently there is an explosion of sequencing of plant genomes. Even there is a project now to sequence 10,000 genomes to cover all the gene families of plants. And for example, in 2019, all of a sudden we got already 3 genomes from avocado. One of them was published in the IHSM. Then in 2020, 2021, we got 3 genomes of mango. And now we are working in the genome of cherimoya that we hope we can publish soon. And there are another annona genome published, like Annona muricata in 2019. And for other of the tropical fruits that we have in our collection, there are also in some cases 2 genome sequence for papaya, 2 genomes for pitaya, 2 for starfruit, and also there are genomes available for lychee, longan, and loquat. But there are not really many tools to work with that. In most of the cases, what you get is just the raw data. So basically, I mean, with the raw data, you can do nothing. You need at least the genome. I would say most of the cases you just get the genome sequences, but in many cases you don't get even gene sequences. You don't get the gene proteins. You don't get the gene models. So there is not much that you can do. And in the cases that you have the gene annotations, the problem is that sometimes they are just a draft. This is the first version, and they didn't have much expression data to validate these genes. And for example, this is the case of avocado. If you go to Koji and you download the data, what you get is a sequence like that. If you know the format, the FASTA format, you might know that the identifier of the gene is just after the greater than, and it ends after the first space. So in this case, all the genes are named per se, and that's the idea of all these genes. So you cannot use it. If you use this file, it will not work in any bioinformatics tool. So the first thing you have to do, you want to use that, is to change the names. I try to change also to the maker names that they have here in the file, but in some cases it's also very long. So some tools such as BLAST might complain. So we need to find a good nomenclature for the gene models, and we need to improve the annotations. And in the future, as much more data are being available in the last year, there will be new versions of the gene models. So my idea is to create some genomic portals where we can host all this data, and we can improve these gene annotations, for example, with web tools such as Web Apollo, where you can manually create the gene models. For that, I'm developing now the IHSM Subtropicals DB, where we will host some of these subtropical fruits that are interesting for us. Right now, we don't have all these fruits. This is a private portal right now, and I just included the data for Annona cherimoya, where we are evaluating the annotations of the genome. And so far, it's quite useful because we can check the gene models in the genome browser. Some people who are working with expression data can check the results, extracting annotations, getting the sequences, or checking some of them with BLAST. But in our institute, many people are working in subtropical fruits, and they are creating a lot of data. So my idea is to integrate all the data created in our institute together with data provided by other collaborators, and integrate all of them in the genomic portal. For example, we have genomics data from germplasm, for phenotyping, morphophysiology, expression data from reproductive biology. We have also data from avocado form, abiotic and biotic stresses. We have also metabolomics data, and many of them will be produced in the next years. However, some of these species have a larger community, and they have a greater economical interest. So in the case of mango, we already developed a dedicated portal only for mango, that is mango-based, which is already public, and everyone who is interested in working on mango can access the data and the bioinformatics tools there. And the idea is like all the germplasm phenotyping data that we had from our collection will be in the subtropical database, but all the genomics data will be linked to the mango base. And to create the subtropical database and mango base and other tools that I will mention in the next slides, I developed my own system to manage all these tools that I call ECGDB, and that I hope that I might be able to publish that soon in bioinformatics, because we just recently got some reviews that were not too bad, and we replied to the reviewers. So finger crossed, I hope we can get it published there. And this tool is very easy to install, not for all the users, but users with basic knowledge of bioinformatics. For example, all my TFM students were able to install it. And this tool have already templates to customize your genomics portal and many bioinformatics tool. It will have templates for the welcome page, the about us page, the genome page, species page, downloads, etc. We have an annotation database that is very flexible to host any type of annotation. And it provides by default many bioinformatics tools such as BLAST, a genome browser, a gene ID lookup tool, an annotation extraction tool where you can provide a list of genes, another sequence extraction tool for a list of genes. And it is able to manage multiple species and multiple annotation versions. The reason I developed this system is I didn't decide to develop that by scratch. It's just, I mean, some other systems are very complex to develop a genomic portal, to develop some web tools. And I wanted to have something very, very simple, very easy to install, very easy to maintain. And as I was already involved in many projects where it was requested to provide a genomic portal, such as, for example, the pre-patents GML-DB, the Olive3DB or ETHNM Arabicum database, I started to create all this code, and then I integrated all together in this new system, so also other people could use it. Now that I mentioned the ETHNM Arabicum, I would like to stop here and mention in a couple of slides this model, because I think it's quite interesting. I don't know if many of you already know ETHNM Arabicum, but when I learned about this plant, I was shocked, because I think it's something impressive. I think it is a great model to study fruit dehiscency, fruit dispersion. And this is because this plant has two types of fruits simultaneously on the plant here. These two fruits are, there's one that is bigger, which is dehiscent, and inside eventually will release the seeds. Inside have from 2 to 6 seeds, which are mucilaginous. So they get sticky with its own humidity. And on the other hand, they have another type of fruit that is smaller and is an indehiscent fruit, which will not open. And it's smaller and only have one seed inside, which is a little bit bigger. The fruits have wings, so they can maybe disperse with the wind or with the water current. So they have different dispersion strategies. And on top of that, the plants show some plasticity, so they can change the proportion of these two types of seeds depending on the environmental conditions. And there are two well-studied ecotypes with a genome already sequenced and with a lot of data. And these are the Turkish ecotype and the Cyprus ecotype, which have the feature that they respond to light differently during seed germination, which is quite interesting. And additional on top of that is ETHNM Arabicum is in a very interesting taxonomic position, because it is the sister branch to all the other Brassicaceae. So it's quite interesting also for the evolution of the Brassicaceae, and it's part of the group of the model plant Arabidopsis thaliana. So I just wanted to let you know, just in case you didn't know this plant, because I found it very interesting. And now I go back to the system I developed to create simple genomics portal in an easy way. How it works is like, for example, in one single file with the configuration path, I can manage multiple projects. In here, I just have the path to for example, to the Subtropical database, to the Mango base, and I just can enable one or the other. And this will take you to the configuration file or each one of this implementation, where you can control the path of all the files on the database. And you can customize everything, and you can control which one of the tools you want to display or not. You can easily enable them or disable them just using zeros or one in the configuration file, just like a switch. And as I said, I mean, you can easily install them. You can use Docker, for example, if you want, and have that in a laptop, in a Windows or a Mac computer. And that way you can have your own data. You can also manage multiple projects simultaneously, and it can host multiple versions and multiple species. Now I would like to show you some of the features that this genomics portal have by default. You just install that and you will get access to most of these tools. It has a downloads section where basically there is a downloads folder that wherever you put there, it's like all the folders or subfolders that you put there, they will be automatically shown in the website with a nice view where you can fold and unfold the sections, and you have links to all the files. And now this is, in my opinion, nicer than the typical FTP links that you see in other sites where they look not very nice. And also recently the web browsers such as Firefox, Chrome, they stopped maintaining the access to FTP sites. So the FTP sites are no longer working anymore. So you need to find an alternative. And I think this one is quite easy because you just can add or remove files and automatically they will be shown in the website. We also provide a search engine where you can search by any keyword of the annotation, or you can search for the gene ID or part of the gene ID. This will show you a view like that, where you have access to a link to all the genes. You have all the descriptions. You can download the data in multiple formats. You can sort the columns. You can search inside the results. We also provide by default a genome browser based on JBrowse. It looks like something like that, in case you haven't seen this before, where you can see your genes there with the exons. You have the introns. You have the UTRs. You get a description. You can load as many tracks as you want, where you can see, for example, expression data, or in this case, these are SNPs. And this is an example of what you can find in MangoBase. And here in this view, we have an example of what we are using in the cherimoya genome browser, where we already have our genes and we can evaluate their annotation. And all these genes are also linked to the gene view. So when you click there, you will find all the information about these genes. The tool also provides an interface for BLAST that I wrote, where you can just add your nucleotide or amino acid sequence, and they will be automatically detected. And we don't need to have a big relational database to manage all the BLAST dataset. Basically, again, we have a folder called BLAST DBs, as you just need to include there your BLAST dataset, and automatically they will be shown in the input view of BLAST. And on top of that, if you have many data sets and you want to put them in categories, like here, for example, by species, you just need to include folders with species and put your data sets inside, and automatically they will be shown. Additionally, if you want to have links to external databases, we have a very simple JSON file where you just provide the name of the data set and the link, and automatically all this new data set will be linked to the external database that you want. By default, the genes will be linked into the gene view, but also in this case, I use, for example, this JSON file to add a link for the genome dataset. And when you find a region in the genome, as most of the times it doesn't have to be inside a gene, then the results is linking you to the genome browser, where you can see actually that region of the genome in the genome browser with all the tracks that we have here. This is how the BLAST output looks like. You can download the results in a tabular format if you want to keep them to format them. It have this classical view with showing the query length with different colors based on the score. It has an additional ID percentage column on the right. It has also a table here on the bottom with the most important information and links to the genes to the gene view. And of course, below you have also the alignments in case you want to see more information. And one of the things of this BLAST implementation is that I try to make it very fast and efficient. So I try to run everything in the RAM memory of the computer, and that way I didn't create any temporary files, so I didn't have to delete later these temporary files. In some other implementations I have seen, it's like they have to write and read for the disk in each one of the steps, which is the bottleneck in the calculation time. So it makes things much slower, and also it creates some temporary files that then you have to have a script to delete them eventually. Another important tool is a tool where you can just provide the list of genes that you want to get the sequence. Then you select the type of sequence that you want to have, and you can download all the sequences. That way you can use all the sequences in another tool that you might want. Again, to control these datasets, they will be automatically be taken from the BLAST dataset. So you don't have to control any relational database. It will be very simple. You put the dataset there, and automatically you can extract sequences from them. One of the tools that I find the most useful ones is the gene extraction tool, where you can just provide a list of genes, and then you can get a lot of annotations for them. And you will get links to these genes to their annotations. You can search, you can sort, and you can download these annotations in many formats. For example, you can download that in Excel. And that is something that most of the researchers use. I've seen in most of the cases, for example, when you have a differential expression analysis, at the end, what you get is a list of genes that are significant in one condition or in the other, but you don't know what these genes are. You just have the gene identifiers. So the first thing you want to do is to get the annotation for these genes. And with this tool, basically you copy your genes, you put it in the tool, and you can just copy them and paste them back in your Excel. So in a couple of clicks, you have all your genes annotated to start discussing about your results, which I find very, very useful. Then I mentioned before many times the gene view. It's like when you click in the gene in the genome browser, when you get the results from BLAST, the results from the search, the results from the annotation extraction, you get links to the gene. For this is how the gene view looks like in this system, in the ECGDB. So at the top, you will have your gene name with the species, the version. You will have a frame to the genome browser to see the gene in the genomic context. You can also get a list of all the annotations with links to their databases. And at the bottom, you can see also the sequence of the gene and the protein. And all these sequences, again, they come from the BLAST datasets. You don't have to control anything else. You don't need to have to upload all these sequences to a database or something complicated. Another nice tool is, like, as I said, many of these gene versions are in the first version and they are like a draft. They need to have a lot of improvement. And in many projects, as these gene versions are improved, you get multiple gene versions, and sometimes you get publications in one version and you have results in another one, and you want to compare between the different gene model versions. Or sometimes you might have some orthologs. So here in this tool, you can provide any kind of list, and you can compare genes from one list to another one, which I find also very useful. So with that, this is the last tool I want to present in ECGDB, which is the simple expression atlas. And I consider, for example, mostly I work in non-model species, and in most of the cases, it's very hard to find expression data for non-model species. Usually when they are published, they just remain as raw data, and you cannot access to that information, which is, I think, essential to design your experimental conditions or to discuss your results, because sometimes you have a gene family and you want to know which gene is expressed in the tissue that you are studying or in the conditions that you are studying. So again, I implemented this expression atlas inside the genomic portal with ECGDB, trying to make things very, very easy. So there's not much work. We can add as many data sets as you want in a very simple way, and you can modify them. So you don't need to have a very bioinformatic high skills, or you need to spend a lot of time on that. So again, we have just a expression data folder, and you just put your file there with a typical format with a tab delimited file with the genes in the first column and the expression data normalized in the other columns. And automatically, this data set will be available already in the input of the expression atlas. Here, we have also a feature where you can just paste a list of genes, and you can get the expression. But if you want to type the genes one by one, you can start typing, and the tool will identify the identifiers in the data set, and automatically will help you to autocomplete that, which I think is very useful for example, for metabolomics data. This tool is also very interactive, so I will show you an example. Here you can choose your data set, and of course you can provide a list of genes. In this case, we have 10 genes. If I want to add another one, I can add the 11th one, for example, and I add it to the list, and I can get the expression. And there we will get many visualization methods to explore this data. For example, these lines to compare the expression of multiple genes, where you can click in the gene name to remove or add genes from the list, and then the scale will adapt to the remaining genes. You can highlight the genes that you want to see also. We also have a heat map where you have multiple ranges of expression that you can highlight to see the different genes in the different conditions. And the idea is, like, this is just the first version. I will add many more visualization methods. In this case, we have also multiple palettes of color. We can download the data in multiple formats. We can also explore the replicates, how they behave in each one of the genes, if they are close to each other or they have a higher dispersion. And at the bottom, we have also a table with all the average of the expression data that you can download in multiple formats, or you can filter the columns you want to see. Again, you can sort. There are multiple pages if you have more than 10 genes. You can search within the table, for example, the genes starting with a 1. And ideally, I will add more and more tools to this, and just adding your data set, you can get access to all these methods. So taking advantage of that, I will try to develop expression atlas for some of the species I'm working on. So I want to create soon an expression atlas for olive and another one for mango that I'm developing with collaborators. And I will try to publish them in a special issue I'm editing together with Professor Gonzalo Claros. And for that, I have the help of some master students. In this case, for Olive Atlas, Amanda and Alier are working on that. And we got also some nice data from collaborators. So in this first version, we are including only the data sets from the variety Picual of olive. And we already have some nice pollen tube growth time series from some collaborators. We also have some nice seeds data from collaborators that are not public, and we will include for the first time in this atlas. And these master students already collected all the public data in repositories, in public repositories, and we already have a few conditions for the expression atlas, such as fruits, leaves, flowers, roots, stem, meristem. We also have abiotic stresses, such as cold or root bounding. We have biotic stresses, such as infection with verticillium. And in the future, of course, we will add other varieties and other data sets that we are already working on. For the mango expression atlas, the master student Ainoa is working on it. We already collected many public data for that, and most of the data are based on the fruit and the peel in the development of the mango fruit and the ripening. So we have data for many of the varieties of mango, and we have some time series also for the storage of the mango and for treatments with hot water brush for the peel, and also we have some time series with pathogen infections. And just to summarize, basically with ECGDB, I already created some tools such as Mango Base, the Olive 3DB, and Ethionema arabicum DB. But now I'm planning to create this expression atlas I mentioned, the expression atlas for mango and olive atlas. And also I have, as I mentioned before, the IHSM Subtropicals database, where I want to integrate all this data that we produce in our institute. And I'm collaborating also in other projects where we're developing a database for avocado, another one for Chlorella ohydrae, for coconut, and also an skeletal atlas for mouse and human. For people interested in this tool, I have all the manual, all the steps to install it and to customize and use it in GitHub. And I also created a reproduction list in YouTube where I present the tool and I mention how you can customize that and which ones are the bioinformatics tools that you can find there. And with that, I would like to thank everyone who participated in these projects, and of course the funding agencies. And I would like to thank you, all of you, for your attention. Thank you.
Well, thank you very much, Noé Fernández, for your talk. This was a great and technical talk that I think it's gonna be, the people working with genomics is going to be enthusiastic. So we have right now in the YouTube chat, we have one question from Xavier Picot, and he wants to know, he's interested in knowing, he said that you mentioned in the problem of poor annotations. Yes. What are you doing to get high quality annotated genomes? He says, I know this is a long shot and might take a lot of time, but needed to make progress.
Yes. Hello, Xavier. Of course, now the thing is, most of this first annotation, the draft, is like they actually didn't have almost expression data when they created this one. So they are based in initial programs, that detect more or less the structure of the genes. And usually they usually sequence a few tissues and they get the expression of these tissues to support the gene annotations. But now it's like, as we can get more and more data, we have also more species that are sequenced. We get proteins of closely related species. We can use all this information that is generated to improve this. It's like usually you have a software that basically runs all this set of tools where you get ab initio software, you get, they check the expression data, the proteins, you can get the repeats, you get a lot of information, and that way you can improve the gene models that you find. Of course, the more expression data that you have, the more chances you have to find genes that are expressed in these experimental conditions that you might not have before. It's like if, for example, I don't know, if you only have the genes expressed in the leaf, when you add tissue from the flower, you might find new genes that are not expressed in the leaf, but they are in the flower. And for that is, I mean, there are several ways to do that. You can have these tools, as I mentioned, where you can manually create these genes, but these go very slow and require a lot of involvement of the communities to improve the genes. And on the other hand, we have this basically automatic pipeline where you can provide all this data and they will generate a new version of the automatic annotation. And that's the idea. I guess that the more versions we get, the better genomes we get, the more expression data we get, we will start to release new genome versions and new gene versions. But it's something that it's an effort of the whole community of this species, and in each case have its own, basically, I mean, you have to involve a lot of people. So a lot of people have to agree with the strategy to follow.
Okay, thank you very much. There are several people popping up with congratulations for your interesting talk, Noe, and also people giving applause with icons. While we have more questions, I think maybe Christoph do you have any question here?
Yeah. So really interesting talk. I think it's, probably underappreciated how much work goes into creating a platform like that. So congratulations. I mean, during your talk, I was playing around with the Mango base and yeah, really nice stuff. What's your target audience for this? Because it's definitely kind of people that have experience with bioinformatics, but that perhaps prefer a GUI version of things, right?
Yeah, I mean, for most of the tools I presented, they are tools that is like any user can use. But for the ECGDB, this is more, I would say, for something with some basic knowledge of bioinformatics. Yeah. But if you want to, in many cases, if you want to maintain that, or even you want to have a local version, I will say if you use any of the other available systems, they are much more complex. So they are much harder to use if you don't have any experience. In this case, I can tell you it's like the master students I have. I mean, I gave them the instructions and they were able to follow, and then they have, basic knowledge of bioinformatics. Of course, you need to know a little bit, and the more you know, the more things you can do there. But with a basic knowledge, you can have your own local implementation and put your data and use these tools.
Yeah. So then, I guess I have a somewhat of a follow-up question on that. What's your opinion as a developer of something like this? Because there's definitely a lot of these initiatives to create these kind of platforms. And sometimes I wonder are you planning on implementing your workflow into some of the kind of scripting workflows that are, I feel like, taking off, like Nextflow or SnakeMake, to kind of help unify the bioinformatics community? Because at the moment it feels like every research niche has their own set of tools,
Yeah, I mean, for this one, this is quite actually a very simple code. So mostly it's based on PHP because it's what the servers have. So there is not a big calculation behind to adapt that to these frameworks that you mentioned. Of course, I mean, this is in Docker and you can integrate that in anything. I tried to remove as many dependencies as I could, because the philosophy I have here is like, I wanted to simplify it as much as I could, because the problem on the long term is it's very hard to maintain this, and you need that is simple enough that almost everyone could maintain that. And this is basically for non-model species where you want to have all these important tools, but of course you don't have a huge amount of data and a huge community behind that. Of course, I mean, if someone is interested, I could integrate that in, but this is not like a pipeline for analysis. This is in ECGDB. Yeah. I mean, for the other ones, of course, there are some algorithm behind, but most of these tools have not really much calculation behind.
Yeah. Okay. Yeah. No. Really cool. I had.
Oh, sorry.
No, go on. I was.
I was more curious on this relationship between the genes and the phenotypes and their expression. Do you think it's possible? I am an ecophysiologist, so I see these things pretty much as a black box from where genes appear that are related to phenotypes, but I don't see pretty well how this happens. So do you think it's gonna be at some point possible that by having a very well annotated genome for a genus of a species, for example, to tell the expression of the phenotype from the genotype? Like if you have this copy or this number of copies from these genes, then you will have this time of ripening, or or this thermal tolerance, which is the trait that I like the most.
I mean, I probably in the future, as we have now a lot of machine learning and things like that, as far as, I mean, if you have defined what are the genes that define one feature in the phenotype, for example, as you said, I don't know, the color of the fruit or the size or whatever, maybe they are able to, with all these markers, with all these genes, to predict some kind of phenotypes, at least for some features. I guess that's what they do with many of these breeding programs. It's like basically you get all the markers that are associated to these genotypes that are associated with the phenotypes. And that way they can predict that they will have all these features. So I guess it's possible, but I mean, I didn't go that far in this kind of data, because as I say, these are non-model species. So usually you don't have all this huge amount of data for that. But I know, for example, for other projects, like they already have a lot of genotyping and phenotyping data that they could link, and probably, I guess you can predict that, at least for some features.
Okay. Another question that I had is so do you think, I was commenting this with Chris the other day. So do you think that for these phenotypes, sometimes complex phenotypes, like for example, the ripening processes, what makes in the case that I'm most interested, which is thermal tolerance, for example, the ability of an individual to survive a given level of temperature, do you think that the genes that code for such a trait that seems a bit complex could be located in a specific parts of the genome, or are they going to be like some sort of randomly distributed across the genome?
Yeah, I guess that will depend on the species. Of course, for some cases there are QTLs where there you have cluster of these genes, but I will say in many cases it could be very complex, and it's not just one single gene or a few genes that control that, because I mean, for this temperature, you might have many things related with the cuticles, with the, I don't know, with many things, with so many things. It's like also with the light, how you get the light, how you can get the intensity of the light, the things that, all this photosynthesis, all the light that you cannot get, the photorespiration. There are many things that probably the plant have to control. Also in the levels of the tissue, the stomas, I don't know. I don't think it's just one single mechanism, but, and also I guess in most of these cases should be very complex. Of course, there might be QTLs where you can get some cluster of these genes that are together in the genome.
And in the light of that, when people, sometimes I'm afraid of making a mistake here because it's really outside of my area. But sometimes I feel that people say, well, we have sampled the whole genome of a species. And we try, so in this way we are kind of out, we can estimate differentiation without incurring in the problem of selection acting on our markers. Do you think it is possible to actually sample a wide array of neutral genes that have not been subjected to selection? Because it feels difficult for me to actually believe that, like, sample a lot of genes and none of them are being subjected to any kind of adaptation process or selection process.
Well, I guess, I mean, it depends, because some of them have a very strong mechanism to maintain them the way they are, because they are essential for the living of the plant. So they might not change. Then there are many studies where you can see all the synonymous and non-synonymous changes, and that way you can predict if the gene is actually changing or not. And it depends, because in plants, sometimes you might have polyploids, and then, or whole genome duplications, you might have many copies of the same gene. And in some cases, one gene will remain stable and the other one will be incorporating or mutations to eventually will change its function. So, I mean, based on these studies of synonymous and non-synonymous, there are some that you can classify as neutral, that is like you cannot see that it's actually accumulating these mutations, or others that are moving to change their function.
All right.
But I think it's possible, I guess, based on that analysis.
All right. Thank you very much. Xavier Pico is also explaining that you need experimental designs, including the entire development of the plant, and circadian rhythms to catch all genes expressing in an organism.
Of course, yeah. It's like if you want to identify all the genes that are expressed, it's like some of them will express in very specifically conditions. So if you don't have that, I mean, many times you can predict that basically based on what you find in the genome. But if you have the support of the expression data, it's the best to get a better annotation.
Thank you very much, Noe, and also Xavier. Very well. As we are approaching the end of the time for questions, do we have any other one in the chat, or Chris?
Maybe Teresa. I think I just saw her hand go up in the Zoom.
Here's our surprise. No. Welcome, Teresa. Welcome.
Thank you. I don't know if you can hear me. Can you hear me now?
Yes, I can see you.
Okay. Nice to see you, even if it's just online. Thank you for your talk. I think it's been great. There are so many resources that you are creating, and I think that's really important. I had a question regarding the VIGS tool. So if I understood it correctly, it looks like you can silence specific genes from the genome just creating small interfering RNAs, and then degrading the mRNAs that are created from those genes, right? Could this be used in crop improvement, for example, and avoiding the problem with GMOs, with genetically modified organisms? I mean, do you think that this could be implemented that way?
No, because usually it's like a transgene also, because you have to introduce your construct is like a transgene. So it's already like a GMO. It's not changing the genome itself. It's at the level of gene expression or transcriptomics, but you are actually introducing an external gene. So I guess that counts already as a transgene. So.
Yeah, I was wondering whether that could be seen not as bad as modifying the actual DNA molecule of the organism.
I don't think that, because at the legal level, I don't think they care, because it's like the new genome editing, which it doesn't matter how good it can get, because at the end, I think it's a legal thing. It's not really a scientific thing.
Mm-hmm. Okay, thank you. And so this is more a comment than an actual question, but I was impressed by all the people that are working in your institute in different things related with the crops, like phenotyping, genotyping, analyzing metabolites, etc. And it looks to me like you are like the glue, trying to put together all the pieces of information and generate resources to actually improve crops. Are you guys, or maybe are you already working with people in agriculture trying to improve these crops, or this still something that needs to be implemented?
Yeah, actually, I mean, the group I joined, they have already an extensive experience on that, on transferring the results to the agriculture. It's like the institute actually was the responsible of introducing the subtropical crops in southern Spain. So they are actually in contact with the agricultures.
So they have been working for years.
Yes,
trying to improve it.
And they introduce also the rootstock. It's like many of the things come through the institute, I would say, and they have a lot of experience. I just joined last year, so I'm trying to help, because as I saw that they are doing so many things, I would like to try to put all together so we can get all available.
Mm-hmm. Great, thank you.
Okay, nice to see you, Tere, and congratulations on your new position.
Thank you.
Bye.
That's great. Thank you very much, Noe and Chris. I think we are reaching 4 p.m. 2 p.m. So I'm gonna finish this seminar. And thanks for all that they're watching. And see you next week with the last seminar of the semester. Bye everyone. Thank you.
Okay. Thank you everyone. Bye.